Today, we are diving into one of the most incredible breakthroughs in modern medicine. We're talking about turning our own immune cells into a living, breathing drug that's engineered to hunt down and destroy cancer. It's a story about biology, high-tech engineering, and just unbelievable precision. So let's just start with this one powerful idea. What if the most personalized, most potent medicine you could ever possibly take was already inside of you, just waiting for the right set of instructions? That is the huge question behind CAR T therapy. This isn't about some chemical you swallow, it's about upgrading your body's own biological hardware. Okay, let's break this down. At its very core, CAR T therapy is this cutting-edge form of immunotherapy. We take a patient's T cells, the natural soldiers of the immune system, and we basically send them to a high-tech boot camp. In the lab, they're genetically re-engineered so they can grow these special receptors on their surface. They're called chimeric antigen receptors, or CARs for short. And these new receptors, they act like a super advanced GPS, allowing the T cells to lock onto cancer cells and, well, take them out.
All right, so here's our roadmap. We are going to follow the entire incredible journey of a CAR T cell. We'll start with the big idea of these living drugs. Then we'll get into the nitty-gritty of designing the genetic blueprint, building the viral delivery system, actually upgrading the T cells themselves, making sure everything is totally safe, and finally, watching them go on their cancer-killing mission.
So first up, this whole concept of living drugs. This is really the absolute heart of personalized medicine, where we're not just treating a disease, we are literally re-engineering a patient's own biology to fight it from the inside out. So here's the most important thing to get. Your T cells are already fantastic soldiers. They're constantly patrolling your body looking for threats. The problem? Cancer is just notoriously clever at hiding from them. What CAR T therapy does is it gives these soldiers a massive upgrade. It hands them a brand new, state-of-the-art targeting system that basically makes cancer cells light up like a beacon in the dark. So how is this upgrade actually built? Well, that brings us to our next section, designing the genetic blueprints. Because before you can upgrade a T cell, you have to write the code for that upgrade. This is where the genetic engineering really begins. The instruction manual for this upgrade is this tiny, circular piece of DNA called a plasmid. You can think of it like the software package. It contains the exact genetic code for that chimeric antigen receptor, the CAR, that we want the T cell to build. So everything, and I mean everything, starts with getting this blueprint absolutely perfect. And what's really fascinating is just how good scientists have gotten at making this whole process super efficient. Using some pretty advanced tech, they can now build these plasmids in a modular way, kind of swapping pieces in and out. They optimize key genetic features, like the part that tells it to start copying itself, and its resistance marker, to make the bacteria that produce these plasmids work way harder. And the result? They get almost a 3-fold increase in production. That is a massive leap in efficiency. Okay, so you've got the blueprint, but how do you actually get it inside the T cell? That brings us to our next major step, building the viral delivery system. You need a way to package and deliver these instructions safely. And this is where scientists get really clever. They take a virus, in this case a lentivirus, and they strip it of all its harmful disease-causing parts. What you're left with is a hollowed-out, totally harmless cell that is incredibly good at just one thing, getting inside of cells. It becomes the perfect delivery truck, packed with our precious plasmid blueprint. Now, creating these delivery trucks is a really meticulous, multi-day process. First, scientists grow these special packaging cells in lab dishes. Then they introduce our CAR plasmid into these cells. The cells get to work, just churning out our lentiviral vectors, which are then very carefully harvested from the culture media over the next couple of days. And now, for the main event. We have the soldiers, and we have the software update sitting in its delivery truck. It is time to actually upgrade the T cell. Transforming a standard T cell into a cancer-fighting CAR T cell isn't instant. It's actually a carefully choreographed 13-day process. It all starts on day 0, when the patient's T cells are activated. Then, on day 1, the lentiviral vectors are introduced. And then comes the expansion phase, where these newly engineered cells are nurtured and encouraged to multiply into a massive army. Finally, on day 13, they are ready for inspection. That key step back on day 1 has a name. It's called transduction. This is the moment the magic really happens. The lentiviral vector, our delivery truck, fuses with the T cell, and it injects that CAR gene blueprint directly into the cell's own DNA. Just like that, the software is successfully installed.
So, how well does it work? Well, in the process we're looking at, the results were pretty remarkable. A full 77.5% of the T cells successfully got the upgrade and started expressing that new cancer-targeting receptor on their surface. That's an incredibly high success rate for such a complex biological procedure. Now, when you are creating a living drug that is going to be put back into a human being, there is absolutely zero room for error. And this brings us to a critical part of the process: ensuring safety and quality. The FDA's definition of purity here really says it all. The final product has to be free from anything extra. No stray bacteria, no unwanted viruses, no contaminants of any kind. This isn't just a guideline, it is a non-negotiable requirement for patient safety. To maintain this level of consistency and safety, manufacturers use a two-tiered system. They create what's called a master cell bank, which is kind of like the original, pristine source code. It's tested like crazy and then locked away. Then, for actual production, they create working cell banks from a tiny sample of that master. This whole system ensures that every single batch of therapy traces back to the exact same safe, qualified source. And the testing, well, it's exhaustive. They check for sterility to make sure there are no bacteria. They screen for mycoplasma, which is a common lab contaminant. They test for any hidden, unwanted viruses. They confirm the identity of the cells, make sure their genetics are stable, and they even test to make certain the engineered cells won't form tumors themselves. It is a true gauntlet of quality control. So after all of that design, all that manufacturing, and all that rigorous testing, we arrive at the final, most crucial question. Does it actually work? This is the final mission: killing cancer. This right here shows the results of a head-to-head test. The engineered CAR T cells were mixed with the cancer cells they were designed to target, and as you can see, they eliminated up to 80% of them. But, and this is the important part, when they were mixed with cells that did not have that target, they left them almost completely unharmed. So the big takeaway is that this isn't some blunt instrument. It's a precision-guided missile. The therapy is both incredibly effective at killing the right cells and incredibly specific, ignoring the wrong ones. The mission was a complete success. And that success is, as the researchers themselves put it, a significant milestone. You see, every single time a process like this is validated, from designing the plasmid all the way to confirming the cancer kill, it pushes the entire field of medicine forward. It offers real hope where there might have been none before. And this leaves us with a pretty fascinating final thought. We have shown it is possible to program a cell with a new function, in this case, to be a cancer assassin. So if we can do that, what other instructions could we write? What other diseases could we teach our own cells to cure? The era of living medicine is really just beginning.